The ultimate goal of protein engineering is to design an amino acid sequence that will fold into a protein with a predetermined structure and function. Paradoxically, this goal may be easier to achieve its inverse, rather than the solution of the protein folding problem. It seems to be simpler to start with a three-dimensional structure and find one of the numerous amino acid sequences that fold into that structure than to start from an amino acid sequence and predict its three-dimensional structure. So here is a good example approached by the two scholars in Caltech. They just inquired, is it possible to design from the first principles a sequence whose main chain obtains zinc finger fold without a zinc atom to stabilize the structure? You learned in chapter 10 about the classical zinc finger domain, which has two cysteine and two histidines to hold the zinc, which stabilizes this short 28 residue long alpha helix followed by a small hairpin structure. They applied their own computer algorithm to this problem. Briefly, this algorithm searches through a very large number of possible sequences using a fast selection procedures for those sequences that stabilize a given fold. They made 10 to the 27th magnitude of different sequences, allowing this residue at core position and this residue at surface, and then also they just some limited residues at boundary. They didn't use proline, cysteine, and methionine, but they used glycine at very limited position, like Chapt. For each sequence, they just use several different conformations. For each, they just calculated energy for non-covalent interactions made possible. Plus, they just use some values of secondary structure propensity tendency. This virtual library size is 15 orders of magnitude larger than an experimental combinatorial library. A corresponding peptide library consisting of only single molecule for each the sequence would have a mass of 11.6 tons of peptide. The actual search space is, however, even larger since different conformations, rotamers, must be considered for every possible side chains. Optimal sequence obtained, called FSD1, is shown in this table. In comparison with the classical zinc finger, FSD1 has no any similarity to any known protein, including this classical zinc finger. In order to examine whether this sequence gave a fold similar to the template, the corresponding peptide was synthesized and its structure was determined by NMR method. You can see the blue one in this structure, which is a designed peptide, and you can see red one from the classical zinc finger. They are very similar. This result shows remarkable demonstration of the power of computer-based combinatorial design of a protein fold. In addition, this design peptide is the shortest known peptide consisting entirely of naturally occurring amino acids that fold into a well-ordered structure without metal binding, oligomerization, or disulfide bond. The last subject is very interesting subject. In 1994, there was some challenge called Paracelsus challenge with a price of $1,000. Basically asking for any designer of a protein structure who could convert one protein fold into another while retaining 50% of the original sequence. The spirit of this challenge was to take one important step towards solving the folding problem by assessing the fraction of a protein amino acid sequence that is sufficient to specify its structure. On the basis of our knowledge of naturally occurring proteins, this challenge did not look like a feasible project. You may know all proteins of known structure with more than 30% sequence identity, homologous proteins, had been shown to have identical folds. It didn't take that long. In 1997, the Line Reagan at Yale University succeeded in obtaining this goal. Beta structure was converted into alpha helix structure by changing only half of the sequence. They started with the domain B1 of protein G, 56 residue long, which has this kind of structure, 4 beta plus 1 alpha. And goal was to get this kind of alpha helix bundle shown in rope, which has a similar length. The sequence identity between two is only 3, so virtually different sequences. The protein they achieved named the Janus protein, which has 28 residue altered from the B1 domain. Rationale to obtain Janus protein was to use current knowledge about the helix formation and stability to identify and change the subset of the residues that are key determinants of the fold, considering both local and long-range interactions. Here you can see some detailed example. Residues in the B1 domain with high preference for beta-pretty sheet formation was replaced with residues with high preference for alpha helixes, such as tyrosine 2 lysine or leucine 2 alanine. And also, rope has a cold-cold structure so that the hydrophobic residues in A and D position of heptade was introduced. 3. Intramonomer salt bridge between arginine 16 and aspartic acid 46 that is present between the two helices of rope was introduced. And 4. The checking any possible strake hindrance. 5. They checked if hydrophobic core was well packed. In this table, you can see amino acid sequences of 3 proteins. The starting domain B1 and the designed protein Janus and rope. The Janus contains 28 changes. It had a 50% identity to B1, and the similarity to rope had increased from approximately 5% identity to 41%. The red residues have been changed to the same type as present in rope. This one. Blue residues have been changed to the different types. Blacks are unchanged. To check if Janus adopt rope-like structure, they just synthesize corresponding gene and then expressed protein from its gene and characterized. But unfortunately, they could not make a three-dimensional structure at that time. But many indirect data indicate that Janus may be very similar to rope and very different from the B1. In conclusion, it is possible to change the fold of protein by changing a restricted set of residues. Second, it might be possible by just changing a few residues in Janus to design a mutant that flip-flops between alpha helix and beta pleated structures. Such a polypeptide would be very interesting model system for applications including prions and other amyloid proteins.